So I hope you had enough time to at least get to know this website and understand how it works. So we are going to go to the next section, but you will have access to this GitHub page. At least, I can assure you, at least for 2 weeks, but this is supposed to get there for a longer period. So if you ever want to go back to the exercise and redo any of the exercise, if you didn't finish, just you can do it. Save the page. Good. So the next section is going to be, and you can download the Git, I think. Yeah. It's going to be about searching for homologous structures. Okay. So we know that we need to do homologous searches, but why do we need to do this? And I always like to start using this image that has this quote by Dobzhansky. And the quote is, "Nothing in biology makes sense except in the light of evolution." So what this means is that evolution is going to be an unifying concept throughout biology, and it will allow us to explain the hypotheses we are making. So in general, in biology, we are trying to find out how life works. And many times we are trying to also find the relationships between different types of life. So one of the reasons why we need to find homologs is that if we want to do evolutionary analysis, what we really want to do is to uncover the evolutionary history of relationships of different evolutionary units. So we usually use as evolutionary units entire organisms, but the evolutionary unit can also be other things. And this is what we are going to discuss now. And then once we finish doing our analysis, what we want to do is express our hypothesis in terms of a phylogeny or a phylogenetic tree. And this is the image I have on the right. And this is our representation of what, how does phylogeny look like. And then we have here a definition of homology, which says that it is classically defined, as classically defined, it refers to a historical continuity in which morphological features in related species are similar in pattern or form because they evolved from a corresponding structure in a common ancestor. So what do we learn from this definition of homology? Well, the first thing we can see is that in this classical definition, it refers to morphological features, which means things that can be seen, that are macroscopic. And then we are going to study the patterns of these morphological features. And this is going to tell us something about historical continuity because these are corresponding structures. And the reason we can do hypotheses with this is just because they are corresponding. If we don't have corresponding structures, we can't compare them. So we know how to study macroscopic features, and this has been done even before molecular techniques. But what if we want to study the diversity of life that goes beyond the macroscopic world? What happens if we want to also study the relationships between multicellular organisms and unicellular organisms, or even with viruses that are not cells and are not considered to be living, but they are biological entities? So if we want to compare these types of biological entities, then we can't use morphology alone. Morphology is not going to tell us much about the history of relationships of these organisms or entities. So, and these are 3 quotes from a very seminal paper from George Fox and Carl Woese. And this is the paper where they propose that there are 3 primary kingdoms of life. And what they say in this paper is that the eukaryotic cell cannot be directly compared to the prokaryotic cell. And they say this because they already knew that according to the endosymbiosis theory, that the eukaryotic cell is a composite. So it's made of at least 2 cells that have merged and they became one. So it's not possible to compare a prokaryotic cell to an eukaryotic cell that is a composite. And then they also say that they can't draw down the phylogenetic relationships comparing the cells because their properties are not comparable. So they propose to use a comparative approach, a different comparative approach, where they use another type of structures to establish phylogenetic relationships. And that's what they did to be able to specify the 3 primary kingdoms of life. And they were not alone in thinking about how to compare all types of living beings. This is a paper from 1964 where Emil Zuckerkandel and Linus Pauling are asking exactly the same question. They are asking where in the now living system the greatest amount of of past history has survived and how it can be extracted. And the answer they give in this seminal paper too is that in some kinds of molecules, we can find this information and extract it to study it. So they say that in genes, mRNA, and polypeptides, there is more history in the making and more history preserved than at any other single level of biological integration. So this is more a philosophical paper, but it really sets the path for all the molecular evolution analysis that we do today. And this is how we get from the macroscopic world to joining all types of life in a single diagram. So we don't go from macroscopic to microscopic, we go from macroscopic to molecular. So what we do is compare genes and proteins because they are comparable structures, and we can establish phylogenetic relationships by comparing genes and proteins. So in here I have a figure that represents a phylogeny that was calculated from sequences of myoglobin from 5 different animals. So just as we can trace back the history of relationships using the bones of vertebrates, we can take their molecules and we can also give a hypothesis of their relationships. And if you see clearly here, it says sequences of myoglobin. So if you remember from the past block, we saw that proteins have different levels of organization. The primary structure is the sequence, and this was built with the primary structure, but it's illustrated in the tertiary structure of the protein. To be able to compute these phylogenies, we need not only homologs, we need a specific type of homolog, and it is the orthologs that we are looking for. So orthology is the phylogenetic relationship where sequence divergence follows speciation. So if we want to learn about the history of the species, we need orthologs. Because orthologous genes often have, and also another way to know that we have an ortholog is that orthologous genes often have the same function in different species. But this is a consequence. It's not always the case that orthologs have the same function. The other type of homolog that will be informative for our analysis, but will not have the same information as an ortholog, are paralogs. So paralogs originate by gene duplication. So they are also homologs. The problem is that if we trace back the history of an organism using paralogs, we are going to be confounded by the fact that they originated from a gene duplication. But something very interesting happens in paralogs. So whenever we have in a genome multiple homologs, they will always be paralogs. And divergence via gene duplication precedes speciation. So the interesting thing about paralogs is that if we find one interesting paralog, we can make the hypothesis that the duplication of this gene came before the speciation. So this is to recapitulate the difference between orthologs and paralogs. So if we follow the red line, the human histone H1.1 will be in red, and then the chimpanzee also has the same gene. These two are orthologs. But then we also know that this histone gene got duplicated before the divergence of humans and chimpanzees. And now we can see the duplication as histone 1.2 in chimpanzees and in humans. And within the genome of chimpanzees, we can see the paralogs, so histone 1.1 and 1.2, as well as in the genome of humans. So now we know that we can use molecules to trace back the history, the relationships between different organisms. So because we are not constrained anymore by morphological features, we can build a phylogeny like this one that is presented here, from a paper, very important paper that was published in 2016, A New View of the Tree of Life. And this paper, this phylogeny in specific, shows the relationships of ribosomal proteins. So again, these are proteins that using the sequences, we can learn about the relationships of many organisms. Another thing about homology. So many times we will hear the term structural homology, and usually referring to excess structure similarity, but homology is a qualitative trait. So when you refer to similarity, you should say similarity, not homology. There are different things. So homology is like asking if two traits are relatives. Like for humans, we say, is someone your relative? And the answer is yes or no. You don't say, yes, he's 25% my relative. So it's a yes or no trait. You can't be less or more homologous, or you can't be highly homologous. You can be highly similar and still not be homologous. So think about this when you use the word homology. The only question we are asking is if they are relatives. And about structure homology, there's another thing. So you can see two structures that are very, very similar and still not be homologous. So in this paper, what they did is that they took one sequence that was designed by an evolutionary directed experiment, and they designed this sequence to bind a ligand. The ligand was not even this metal ion that is shown in here. It was another molecule. And then at the end of this experiment, this directed evolution experiment, they took the molecule, crystallized it, and showed the structure in here. And they realized that it's very similar to a protein that occurs in nature. So because the protein in panel B came from an experiment that was entirely designed by humans, you can't say that protein B is homologous to protein A. And yet they are very similar in structures. This is showing that highly structural similarity exists to the point that there can be convergence to one shape without homology. So be aware of that when you use protein structure for your evolutionary analysis. What this means is that every time you use a protein, you need to know also a protein structure, you also need to know the similarity in sequence or the similarity in function to make the hypothesis of homology. And your argument is going to be stronger if you have the three of them, if you have very similar sequences with a very similar structure and a very similar function. I know this is not always the case. You may be working with a protein that is only similar in structure, but there are methods that we can use to try to really find if these similarities are due to homology. What we do know how to use is sequence similarity. So these very early works before 2000, this is a review, determined that you can know by the sequence if two sequences are homologous or not, because whenever you find an abnormally large number of amino acids that can be aligned, then you can make the hypothesis of homology. And there are many techniques to do that. The most frequently used one are BLAST and FASTA. Those are two programs that compare sequence versus sequence, and they can give you a result to know if the excess similarity can allow you to make the hypothesis of homology. If you want to use a more sensitive method, then what you can do is take your sequence, the sequence of interest, and compare your sequence not to another sequence like in BLAST and FASTA, but compare it to a multiple sequence alignment. So we don't really compare it to a multiple sequence alignment. What we do is we transform the information contained in a multiple sequence alignment into a profile. And this profile can be just an average of the frequency of residues in each position, or it can be a statistical model of these positions. So there are different ways to construct these profiles, but in the end, what you are doing is comparing one sequence with a multiple sequence alignment. And a more sensitive method is now to compare a profile versus a profile, meaning compare one profile to a database of profiles and find out if you have a multiple sequence alignment that looks similar to another multiple sequence alignment. So, of course, you can use sequence, but then you have to go back and from this, of course, you can use structure, but you have to go back to the analysis of sequences to know what the similarity in structure means in evolution. So you can use servers like DALI or cofactor to find out if you have a similar structure, and then do all the research to try to investigate these relationships. So now we are going to go to our second block of exercises. I think I'm going to share again the link and go to the GitHub page. So at each time, the exercises will get somewhat harder to do, but you will be able to do them just using your web browser.